Hi, good morning. Can you hear me?
Yes.
Yeah, Professor Tsenkova, congratulations.
Thank you.
Yeah, so I'm a chairperson for next session. Sai Tanaka from Keio University and the National Institute of National Science. So Professor Dr. Uchida and Dr. Ishigaki here.
Yes, I can hear you.
Okay, so let's start this session. Dr. Uchida, could you prepare the slide? And
yes.
During it, I try to explain about this session. So this session is worded as a part of biological process. As you know, living organisms consist of more than 70% water, and water is essential for all life on Earth. But our understanding of water is so partially. I think it's because the biological system is quite complex compared with in vital system. In this session, we have two speakers. The first speaker is Dr. Uchida, Associate Professor at the Division of Applied Physics, Faculty of Engineering, Hokkaido University in Japan. His research focus has been on water science such as cryobiology, ultrafine bubbles, ice and gas hydrates. Today, I'd like him to talk about the cryopreservation of cells. In the title, Study on Cryopreservation Mechanism using Trehalose-transporter expressing cells. Are you ready?
Yes,
Dr.
thank you
Uchida?
very much.
Yeah, please start your presentation.
Can you see my presentations?
Yes.
Yes, okay,
Yes.
I will start. Thank you very much, chairpersons. I'd like to show you about our recent activity of cryopreservation of living cells. Unfortunately, we don't have any materials for aquaphotomics, but we are interested in the water, so it's related to the water science. The cryopreservation is a well-known technique to use in several industries, such as livestock industries. It is also the unique technology to stop the time of life. However, its mechanism has not been revealed scientifically yet. Because water is a major content of the cells, everyone understands that it will be related to the phase change of water. So if we control the phase change of the water, we will control the time of life. So our aim is to reveal the cryopreservation mechanism from the viewpoint of the water. When we cryopreserve the living cells, we need to use a cryoprotecting agent, Cpa, to control the phase change of water, or to dehydrate the intercellular water, or to protect the organelle, membrane, and so on directly. Usually, we use the Cpa as a glycerin or a DMSO, but these chemicals are known to have the cryotoxicity. So we are necessary to develop the alternative CPAs, such as antifreeze proteins or sugars. We use the trehalose for the CPA candidate of CPAs because some animals use the trehalose to resist the freezing stress. Also, the trehalose inhibits the ice growing. Unfortunately, the trehalose cannot penetrate into the cells due to its large volumes. Our colleague discovered the transporter, Trehalose-transporter named TRET1 in 2007. So we decided to construct the cell lines to study the cryoprotecting effect of intracellular trehalose. Our objective is the experimental study of cryoprotecting effect of intracellular trehalose by cryopreserving two types of Chinese hamster oval cells simultaneously. The normal CHOK1 cells is showing the effect of extracellular trehalose, but the TRET1 expressing cells can show effect of both intracellular and extracellular trehalose. So our first study is the measurement of freeze-thawing viabilities depending on the trehalose concentration and cooling rate. The experimental procedures is very simple. We culture the two kinds of cells and preparing the cell suspensions. And we replace the culture medium to the freezing liquid. It includes the trehalose from 0 to 1 molars. And the sample is incubated at 37 degrees Celsius for 6 hours to uptake the trehalose into cells. This is a schematic diagram of trehalose uptake in the oocyte in the different cells. But 6 hours is enough to introduce the trehalose into the cells. After that, we freeze the samples at different temperatures to control the cooling rate from 1 Kelvin per minute to 200 Kelvin per minute. And after 1 week stretch, we thawed the samples at 37 degrees very quickly. And changing the freezing liquid to the culture medium to remove the intercellular trehalose automatically. To measure the viabilities, we use the double staining method. We use the fluorescence microscope, and the green light shows the living cells and the red light shows the dead cells. In addition, we cultured the survival cells to check their healthiness by observing the adhesion and the prolifications. This is the result of the viability depending on the trehalose concentrations. The normal cells, the viability of normal cells is less than 10% with very small under all concentration conditions. So we consider the extracellular trehalose has only small cryoprotecting effect. On the other hand, the TRET1 expressing cells, the viability is very high, especially at the 400 millimolars. It's the maximum about 80%. So we confirm the intracellular trehalose exhibits a sufficient cryoprotecting effect. We also check the healthiness of the surviving cells. And these observations confirm that the function of the survival cells, function means adhesion and prolification, is enough according to their viability. 70% is full, the 10% is partly. Another result is the cooling rate dependence. For the normal cells, the viability is lower at all cooling rate conditions. In order to check our feasibility of experimental processes, we use the DMSO 10% freezing solutions in the same experimental process. And these high viability of DMSO solutions show our feasibility. But the TRET1 expressing cells has higher viabilities around the cooling rate is smaller than 50 Kelvin per minute. So we consider that the intracellular trehalose is effective in slow freezing cryopreservation. So we move on to the next consideration. We consider about the freezing conditions during the dehydration time and the trehalose uptake time. We measure the freeze thawing viabilities depending on the immersion time. Also, we check the uptake amount of intercellular trehalose. Based on this result, we discussed about the cryoprotecting mechanism of intracellular trehalose. The experimental procedure is almost the same as before, but we change the immersion time from 0 to 6 hours, here to here, to change the dehydration time or change the uptake amount of trehalose. And these samples are freezing at -80 degrees Celsius and checking the viability depending on the immersion time. And separately, we measure the amount of intercellular trehalose by high performance liquid chromatography. This is a result of the viability of normal cells depending on the immersion time. This figure shows the viability decrease with increasing the immersion time. Previously, I showed the viability is almost 0 in normal cells. It is the immersion time is 6 hours. But the normal cells can survive at shorter immersion time. So we changed our conclusion that the extracellular trehalose can work as a CPA. We also checked the uptake amount of trehalose of normal cells. As expected, the uptake amount of trehalose is very low, independent of the immersion time. So we confirmed that the normal CHOK1 cells cannot uptake the trehalose without TRET1. So we consider the normal cells were weakened during the immersion period before freezing due to osmotic stress by extracellular trehalose. On the other hand, the viability of TRET1 cells, depending on the immersion time, is increasing at lower concentration, lower than 400 millimolars. So we consider the intracellular trehalose exhibits its cryoprotecting effect depending on the uptake amount. At the concentration is 400 millimolars, viability reaches at the maximum, dependent on the time larger than 2 hours. However, the concentration is more than 500 millimolars. The maximum viability is observed in a shorter period. Here is 500 millimolars, 1 molars, shorter immersion time. And after that, the viability decrease depending on the time. So we consider that when the concentration is higher than 400 millimolars, the negative process will be occurred similar to the normal cells. We measured the uptake amount of trehalose in TRET1 cells. And as you see, the uptake amount of trehalose, depending on the immersion time, is very similar to the viability curves. For example, the concentration is smaller than 400 millimolars. Both trehalose uptake and viability increase with immersion time. and 400 millimolars, it becomes the maximum. And larger concentration, the Trehalose uptake and viability also decrease with time and concentration. Thus, we consider the higher the amount of intracellular Trehalose, the higher the viability. But there is a limitation of Trehalose uptake into the CHOK1 cells. As a discussions, we estimate the maximum amount of intracellular Trehalose. The measurement shows the maximum is 80 picogram per cells. Using the effective volume of CHOK1 cells, the concentration is about 480 millimolars per cells. So you can see the saturation is achieved in this range, concentration range, and intracellular and extracellular Trehalose concentration becomes equal. At that moment, the optimum condition is occurred. More than optimal conditions, the concentration is higher than 500 millimolars outside of the cell is always higher than the inside the cell. So the osmotic stress is applied every time. So even Tret1 cells weakened due to the osmotic stress after reaching the saturation. Finally, we discussed about the dehydration effect of cryopreservation using the very simple model. If when we add the Trehalose at the concentration of C0, the cell volume V0 will be changed due to the dehydration about delta V until the concentration difference is finished. Using these models, we calculate the volume change ratio due to the dehydration as these curves. When the concentration is less than 500 millimolars, the viability increase with increasing the intracellular Trehalose concentration. At that moment, the dehydration seems to be almost negligible. So we consider the Trehalose, Tret1 cells are cryopreserved mainly by the cryoprotecting effect of intracellular Trehalose, not the dehydration. On the other hand, the concentration is larger than 500 millimolars. Dehydration occurs depending on the concentration of Trehalose. At that moment, the viability decreases simultaneously. So the dehydration may weaken the cells before freezing. So we are considering, reconsider the effect of dehydration. It is considered to be the very important for the cryopreservation technique, but we need to reconsider, especially for the slow cryopreservation technique. This is the conclusion of our experimental study of CHO Tret1 cells and Trehalose. We found that the presence of intracellular Trehalose greatly improves the viability, but dehydration by osmotic pressure difference may not be important for slow freezing cryopreservations. This is the final slide. Our future plans related to the aquaphotomics. It is my homework, I think. First one is the understanding of the conditions of intracellular and extracellular water. It may be the structurized or what has happened in dehydrations. We can collect the many information in this conference. The second one is understanding of the cryoprotecting effect of Trehalose itself. It is considered to induce the glass transition of water or protect the cell membrane directly. We cannot find any evidence at yet. Maybe our chair will be present tomorrow related to the sugars. Also, we also understanding the dehydration effect of cryopreservation. We can use the aquaporin expressing cells, which is already shown by the Professor Yasui. And if we use these cells for the cryoprotecting studies, we can compare the tryptophan cell expressing cells, and we can observe some information about the dehydration effect. Thank you for your attention.
Thank you for Dr. Uchida. It was great presentation. So any question or comment? We have the 2 minutes left. Do you have any question? No? Okay. So I have one question, okay?
Yelena.
Yeah.
Sorry,
Yelena. Yeah. Okay,
I'm
okay,
sorry.
okay.
I'm sorry, Tanaka-sensei. So
Yeah.
I really, I mean, you said at the beginning of your presentation that you did not do aquaphotomics work yet, but this is absolutely aquaphotomics. It's so valuable to hear.
Thank you.
My question is, so how can you explain this effect of the, I mean, do you have idea what is happening? Why is this the limit for saturation, this 500 millimole? So what is happening before and after in regards to the cell viability? That is my first question. The second question is, because this all talk reminds me of many, many things in not only cell preservation, but food preservation. So there are many different substances that act in a similar way for preservation, like salt. You said different types of proteins and so on. So can you can you suggest there is some kind of common mechanism behind the action of all these substances? Thank you.
Thank you for your questions. It's a little bit big questions. The first one is limitation of the uptake of amount of trehalose in CHOK1 cells. We don't have any evidence for that, but we consider the cell volume is smaller than the other cells. So the limitation will be depend on the cell volumes. For example, when we use the oocyte, the limitation should be larger because the oocyte is a little bit larger. But we cannot check the limitation yet. So the mechanism. I think the uptake amount of trehalose will depend on the time, immersion time. So when the amount of trehalose is small, the cryoprotecting effect will be small. But after that, after the limitation is occurred, the more trehalose cannot penetrate into the cells, but the concentration gradient exists from the inside to the outside. So the dehydration will occur. So we consider the timing is different. First, the trehalose uptake into the cells. At that moment, the water will not move so much. it means that the Trehalose transportation is very quick compared to the water. After that, moving of the Trehalose is stopped. The water start to dehydrate, and it will be shrink. The cell will be shrinked. The second one is very difficult to answer. I think the third my homework using the aquaporin expressing cell will be a very good comparison with this our studies. Can I answer to your questions?
Yes, thank you. Actually, you really answered a lot by this. So I understood. I connect what you're doing with some studies that we did with plants, which are preserved by sugars like this. And for those, also, it is important, the rate of drying is important. So this is what I just now I saw why it is important from your presentation. And now when you said about this limit, I realized the viability, that is something that we also see in preservation of food, that it is about preserving the mobility of water species, not about water content, but this part that is highly mobile. So this was really, really nice to hear. Thank you. Thank you a lot.
Thank you very much. I agree with you.
Yeah, thank you. So, but we have no time left. So the time to move on next speaker. I'd like to thank Dr. Uchida again.
Thank you very much.
So our next speaker is Dr. Ishigaki. Ishigaki is here. Okay.
Yeah.
I can find it. Yeah. And share the slides. So okay, let's start the next speakers. So our next speaker is Dr. Ishigaki, an assistant professor in Faculty of Life and Environmental Science.